Hey guys, this is the first of our videos on blood vessel anatomy. So I'm going to start sharing my screen with you now. And I've got a PowerPoint here. So these are our slides from the blood vessel section. And we're going to first look at the structure of blood vessels. So in the lab, you learned that every blood vessel is made of 3 layers: the tunica adventitia, the tunica media, the tunica intima. So tunica just means layer. So layer adventitia, layer in the middle, media, and layer that's next to the blood, intima. So I often remember that the tunica intima is intimate with the blood, so that means it's close to the blood, right? Media means in the middle, and then the adventitia is the outside layer. So which of these layers is going to have our muscle in it? It's the middle layer, the tunica media, that has the muscle. And what type of muscle do we find in tubes and bags around the body? We find smooth muscle. So our tunica media is going to be the smooth muscle layer, the middle layer of the blood vessel. So let's take a look at this example of a blood vessel. So here's the tunica intima. The blood is here. So remember I told you this is intimate with the blood. So it's right next to the blood. Where you see this line, this squiggly line, this is called the internal elastic lamina. And that's kind of like your dividing line that's between the tunica intima and the tunica media. Got it? So this internal elastic lamina shows you where your division is between intima and media. Now remember we said media was muscle, so here are all our smooth muscle cells. Can you see that they've got the spindle shape that we learned about in Biology 203? So this is our tunica media, and here's our adventitia. The adventitia is the connective tissue layer that's going to connect this blood or this blood vessel onto the surrounding tissues. So remember, all around blood vessels, you're going to have muscle, other connective tissues, bone. So to glue this blood vessel in place and hold it in place, that's the function of this adventitia structure. So it holds the blood vessel in place. Also, in really thick blood vessels like the aorta that have a really thick muscle layer and really thick walls, there are tiny little blood vessels in this adventitia that help to nourish these cells. So the vasa vasorum are these tiny blood vessels that help nourish the walls of the huge blood vessels like the aorta, the vena cava, these really large blood vessels. Because remember, muscle cells need blood or they need oxygen and nutrients to stay alive, just like any other cell. So in a really thick muscle layer, in a really thick blood vessel, these middle guys would be too far away from the bloodstream to get any nutrients. So that's why we have the second round of blood vessels in the adventitia to help nourish these muscle cells because they're going to need a lot of oxygen and nutrients. So we understand that there are 3 layers: tunica intima, tunica media, tunica adventitia. Another important structure is right here. This guy right there. That is called what? It's the lining of a blood vessel. It's a type of epithelium. It's called the endothelium. Okay, so this is a layer of epithelium that lines the blood vessel. So the blood would be here. Here's my epithelium, and then we're into the wall of the blood vessel here. Okay, so let's learn a little bit about each one. So like I already told you, our tunica intima is intimate with the blood. So it's right next to the blood, right next to the lumen. Remember, the lumen is the space that the blood is going to be inside. So the blood is here, right? And here's my endothelium right here, this lining of the blood vessel. So the endothelium is a really important layer. It doesn't just act as a kind of barrier between where the blood is and where the muscle is. It also is very active. It can produce all type of chemical signals that can either relax or contract blood vessels. So the endothelium is a really important part of the blood vessel. It can send lots of signals and tell this muscle to contract and relax. Under the endothelium, you have some areolar connective tissue or packing connective tissue. It's basically just gluing the endothelium onto the muscle that's underneath. So let's have a look at some of these chemicals that endothelial cells can produce. Nitric oxide is a big one. So the endothelial cells can make this. Very important, nitric oxide relaxes blood vessels. Prostacyclin, another chemical that comes from endothelial cells. Prostacyclin relaxes blood vessels. And then our third one, endothelin. This is different to the other two because this one's able to contract our blood vessel. So depending on the situation, if you want to relax your blood vessel, make it larger, make it easier for blood to flow through it, you can signal to the endothelium to produce these guys, nitric oxide and prostacyclin. But if you want to constrict your blood vessel, then you're going to tell your endothelium to make endothelin. So it's very important that you know which of these is a relaxer and which one is able to contract blood vessels. Okay? Now let's look at the muscle layer, the tunica media. So the tunica media, remember in bags and tubes, we have smooth muscle cells. So we know what they look like. They're long, kind of spindly cells like this. The nucleus is kind of in the middle and cigar-shaped. And it's a big sheet of cells that are all connected to each other. Now, some of your big blood vessels, every time the blood flows through, they have to stretch, and then they have to go back to normal size. So I might actually just come out of this for a second just to give you a visual on this one. So let me go back here and just show you quickly. So when you have your blood vessel, blood's flowing through it. So for example, when the heart squeezes all that blood into the aorta has to expand as the blood comes in, and then it has to contract to push the blood forward to the next area. So your aorta is going to be constantly going like this all through the day. Every time the heart beats, your aorta is going to be going in, going out, going in, going out. So that's what gives you your pulse. When you're putting your hand on your wrist here, trying to feel a pulse in somebody, you're feeling the blood vessel here, the artery here, doing this every time the heart beats. So you're counting how many pulses you feel. Every pulse is one heartbeat. So let's go back to our PowerPoint and see this again. So when we're in this tunica media section, we've got lots of elastic, especially in the big arteries. They're constantly pulsing every time the heart beats, and every time a rush of blood is coming through, they have to stretch, and then they bounce back to normal size. So there's a lot of elastic tissue. So remember, blood vessels can either constrict or they can dilate. So here's my blood vessel normal size. Okay. If it constricts, getting smaller. If it dilates, it's getting larger, right? So let's label these guys up. So if it gets smaller, that's vasoconstriction. If it gets larger, that's vasodilation, right? Dilating, getting larger. So it can either constrict or dilate. The sympathetic nervous system usually constricts blood vessels. The parasympathetic nervous system usually relaxes blood vessels. But there is some exceptions to this, and we're going to talk about those in class. So just like with the heart speeding up and slowing down, sympathetic, parasympathetic, with the blood vessels constricting or dilating, sympathetic, parasympathetic is going to affect there as well. So that's our muscle layer. Now next up is our adventitia, the connective tissue that glues the blood vessel and holds the blood vessel in place. So there's nerves running through this connective tissue, lymphatic vessels running through this connective tissue, and small little capillaries and blood vessels running through this connective tissue too. So remember, if you have a really thick-walled blood vessel with lots of muscle, so all of this is the media, so all of this is the muscle layer, the blood is going to be way over here, right, in this section. So these guys in the middle, how are they getting oxygen and nutrients? So in the adventitia, these little capillaries and blood vessels, are delivering the oxygen and nutrients through to these muscle cells. So the small blood vessels that nourish larger blood vessels is called the vasa vasorum. So our adventitia is protecting the blood vessel, reinforcing the blood vessel, and holding it in place, attaching it to the surrounding structures. So let's compare an artery to a vein. What is the difference between the two? Arteries are under a lot of pressure. These guys are receiving blood from the heart, so they're going to have really tough, thick walls. So they have a thick tunica media. So that means that they have lots of smooth muscle. So under the microscope, the wall is going to look really thick because it's going to have lots of smooth muscle. The adventitia, the connective tissue layer, is very thin. You never ever find valves in our arteries, and the lumen under the microscope, an artery is going to look perfectly round. Because remember, it's got that thick muscle wall. So remember, to prepare a slide for the microscope, you have to chop up your blood vessel and slice it into little thin slices. And because this has a nice thick muscle wall like this, it's able to withstand that cutting process, and the arteries are able to maintain their nice round structure under the microscope. Now veins, on the other hand, have a very thin tunica media, so they have very little muscle. So what happens to them when you try and prepare them for the microscope, because they have such a thin muscle layer, they kind of get collapsed and squashed. So they no longer look round, they got this kind of squashed appearance to them, and the wall is really thin. Also, veins have valves. So every so often you might see a valve, a little flap of endothelium sticking in here. And remember, we learned that you push the blood through the valve, and now if it tries to go back here, what's going to happen? It's going to close the valve. So valves prevent backflow. So that's the kind of major function of these valves. They prevent backflow of blood, right? So our valves are going to be really important structures to make sure that the blood in the veins keeps coming back towards the heart. Even though it's fighting against gravity and all these things coming up from the legs, it has to keep coming in the right direction. So remember, valves in the heart help keep blood going in the right direction too. So let's look at our picture under the microscope. So this one is labeled already, but if you didn't have these handy labels, would you be able to tell who was the artery and who was the vein? So remember the clues I gave you. I told you arteries have a really thick muscle layer. So can you see that the wall of this artery is this thick? That's all muscle right there. In the vein, this is only the little bit of muscle that we have. So can you see that this is much wider than this? So this is round and sturdy. There's also lots of elastic. All those black things you see are elastic fibers, because elastic stains black under the microscope. So this has got a really thick, sturdy wall. This one's got a thinner wall, and also look at the shape. The artery is still nice and round, right? The vein is totally squashed. Excuse me. So that's how you can tell the difference under the microscope. This is an artery, this is a vein under the microscope. And all the surrounding stuff is connective tissue. Here's some adipose tissue you learned about in Biology 203. These are some neurons or nerves over here. So all of this stuff is kind of close together. But you can tell the difference between the artery and the vein by looking at the thickness of the wall and by seeing who squashed and who stayed nice and round. Okay, so that's the end of our first video about blood vessels. So I'll just go back here. So hopefully you enjoyed this one, and there's a next one coming now with further information about blood vessels.